Good afternoon, class. This is the second online lecture on the structure and function of muscle cells and muscle tissue. So today we're going to talk about the four steps that have to occur for skeletal muscle to contract. So first, there have to be nerve stimulation. So remember, skeletal muscle is voluntary, so it needs nerve stimulation to happen. And this nerve stimulation is going to occur by an action potential traveling from a motor neuron all the way down to a neuromuscular junction. Okay, and the action potential is generated in the sarcolemma. And if you remember from our previous lecture, the sarcolemma is the plasma membrane of the muscle cell, the muscle fiber, and it's going to be propagated along the sarcolemma. And then intracellular calcium level is going to rise briefly. And we're going to go into these four steps in more detail. And it's actually a lot easier to understand how an action potential works or how this process works by looking at figures. And we will go over several figures. But essentially, steps 1 and 2 occur at the neuromuscular junction, and steps 3 and 4 link electrical signals to contraction, and it's referred to as excitation, contraction, coupling, and I will use EC coupling for short. All right, so this figure is showing you phase 1 and phase 2 of muscle fiber contraction. So essentially, phase 1, it's going to motor neuron stimulates the muscle fiber. The one motor neuron can stimulate several to even hundreds of muscle fibers. All right, so an action potential is going to arrive at the neuromuscular junction, and then acetylcholine, that ACH is acetylcholine, is released, and it binds to receptors on the sarcolemma. So essentially, at the neuromuscular junction, acetylcholine gets released into a little space in between the neuron and the muscle fibers. There's a little gap, a little space between the neuron and the neuron terminals, the axon terminals, and the muscle fibers. And in that little space, acetylcholine gets released, and then it's going to bind to the sarcolemma on the muscle fibers on the other side of that neuromuscular junction. Okay, and the acetylcholine binding to that sarcolemma is going to cause a depolarization of the membrane, essentially ion-gated ion channels, voltage-gated ion channels, meaning a change in voltage causes the ion channel to open, and that's going to allow sodium to rush in, potassium to rush out. That's going to cause a depolarization of the membrane. Normally, at the resting membrane of a cell, it is around negative 90 millivolts. So, and you have this sodium potassium pump that's constantly pumping 3 sodium out of the cell and 2 potassium in. So you also have a buildup of sodium concentration outside of the cell. So when you get this depolarization, when you get this voltage changes and allows these sodium ion channels to open, sodium wants to rush in, one, because it's negative on the inside, and also because sodium is concentrated higher on the outside of the cell than on the inside of the cell. So that local depolarization ignites an action potential in the sarcolemma, and then that action potential is going to travel from the sarcolemma all the way down into the T tubules. Remember, the T tubules are an extension of the sarcolemma. And then at these T tubules terminals, you're going to have the sarcoplasmic reticulum It's going to release calcium. All right, that's going to essentially bind to troponin, and it's going to allow the myosin to bind to the actin thin filaments, and then that myosin binding to actin is going to cause contraction. It's going to cause the myosin to slide the thin filaments. Okay, so that's the excitation contraction coupling that's occurring. And again, this is a lot more clear when you see different figures, and we're going to look at different figures so we can see these steps more clearly. Okay, the nerve stimulus and events at the neuromuscular junction. So again, we're going to say a figure of the neuromuscular junction, but remember that you have a motor neuron, okay, and you have the axon is where the signal that goes down the motor neuron, and the axon's going to end at a neuromuscular junction. And essentially, this neuromuscular junction, you're going to have these thread-like extensions of motor neurons, okay, that's going to send its signal to multiple, up to hundreds of muscle fibers, okay. So the axon branches in on the muscle fiber, forming a neuromuscular junction or motor end plate. And each muscle fiber has one neuromuscular junction with one motor neuron. This is the figure of a neuromuscular junction. You see right here, this is an axon, and you see how at the end of this axon it's branching off, okay? So the axon terminal is branching off at the end, and each of these axon terminal is going to make a neuromuscular junction with a muscle fiber, okay? All right, and this is showing you an up close picture of a neuromuscular junction, and you see how you have this little synaptic cleft, this little space from the axon terminal and the muscle cell fiber. Within this space that's where the acetylcholine gets released from the axon terminal, and then it's going to bind to receptors on the sarcolemma of the muscle fiber. That's going to allow a voltage sensitive ion. It's going to allow a sodium channel to open, which is going to depolarize the membrane, and it's going to open up other voltage gated ion channels, and it's going to propagate an action potential. And we're going to talk about more about the role of calcium, but calcium is actually playing two roles. So with this, you have an action potential arriving at the axon terminal of the motor neuron. And then right here, these are voltage gated calcium channels open, and then calcium enters the axon terminal, which causes the acetylcholine to be exocytosed. So these voltage gated calcium channels are in the axon terminal, okay, and they're causing the acetylcholine vesicles to be exocytosed, and then the acetylcholine is going to bind. It's going to cross the synaptic cleft, and it's going to bind to receptors on the sarcolemma of a muscle fiber, which is going to open up chemically gated ion channels, sodium, potassium. So sodium is going to rush in, potassium will rush out. But we will also have calcium that's going to deal with the muscle fiber and the sarcoplasmic reticulum, and that calcium is going to be released from the sarcoplasmic reticulum in the muscle fiber, and that's going to it's going to bind to troponin, and then that's going to allow myosin on the thick filament to bind to actin on the thin filament, and that allows for muscle contraction. So don't get confused. So this calcium on the axon terminal plays a role in causing the acetylcholine to be exocytosed from the axon terminal, but then you can also have calcium channels, the calcium that's going to be released in the muscle fiber through the sarcoplasmic reticulum, which is going to be the last steps of muscle contraction. Okay, so don't get confused. Calcium is just playing two different roles. And the rest of the figure actually has steps 1, 2, 3, where it talks about that. Okay, the action potential arrives at the axon terminal. Voltage gated calcium channels open, and the calcium enters the axon terminal, and it's going to cause the acetylcholine to be exocytosed in these vesicles. So those writing just explains what the calcium is doing in the axon terminal. Okay, we're not talking about the calcium yet in the muscle fiber that's going to be released from the sarcoplasmic reticulum. Okay, this is talking about the calcium, what it's doing in the axon terminal. This slide is just talking more about the neuromuscular junction. So remember again, the axon terminal and the actual muscle fiber are separated by this synaptic cleft, which is just a gel-filled space, okay? And within the axon terminals, you have these synaptic vesicles, which are filled of acetylcholine. And the acetylcholine is going to get released from the axon terminal. It's going to go through the synaptic cleft, and it's going to bind to receptors in the sarcolemma, the plasma membrane of the muscle fiber, and these infolding called junctional folds. Okay, and these junctional folds contains millions of acetylcholine receptors. All right. So this is showing you an additional step. This is showing you the acetylcholine diffuses across the synaptic cleft and binds to its receptors on the sarcolemma. Okay, and remember, these receptors on the sarcolemma are ion channels, and they are going to be sodium and potassium ion channels, and it's going to open up these ion channels. And remember, in a normal resting membrane, the inside of the cell is around negative 90 millivolts, and the outside of the cell is positive. And you also have an accumulation of sodium cells outside the cell because of the sodium potassium pump that pumps 3 sodium out and 2 potassium inside the cell. So when these sodium ion channels open, when the acetylcholine binds these receptors, these ion receptors on the sarcolemma, it's going to open up a sodium channel, and sodium's going to want to rush in, because remember, particles always want to go from where they're highly concentrated to where they're low concentration. That's called down the concentration gradient. So sodium wants to go inside the cell because it's concentrated outside the cell, and also inside of the cell is negative. So remember, those opposite charges attract. The positive from the sodium is going to be very attracted to that negative charge within the cell. So those two things are going to drive sodium into the through the ion channel, and it's going to cause depolarization of the membrane. And depolarization of the membrane just means that the membrane is no longer going to be negative 90. It's going to be becoming more positive. That's what all that means. All right, so this is showing you step 5. Acetylcholine opens ion channels receptors that allows the simultaneous passive sodium to the muscle fiber and potassium out. So also, again, remember, with a sodium potassium pump, it's concentrating sodium outside of the cell, potassium inside the cell. So sodium wants to come in, potassium wants to go out. Okay, those are their natural concentration gradients. Remember, particles always want to go from where they are highly concentrated to where they are lowly concentrated. Okay. So more sodium ions enter than potassium ions exit, which produces a local change in the membrane potential called the end plate potential. This slide is pretty much summarizing what we got through talking about on the figure. So again, you get a nerve impulse, so action potential travels down an axon terminal, causing acetylcholine to be released to the synaptic cleft. It's going to diffuse across the synaptic cleft and bind to receptors on the sarcolemma, which is going to open ion channels, which allows sodium to come in and potassium to go out. And essentially, that's going to cause depolarization of the membrane, which is going to cause an action potential to be generated at that location. And so essentially, you don't want the acetylcholine to stay in the synaptic cleft, because that's gonna, if it stays in the synaptic cleft, it's gonna be constantly opening up the ion channels in the sarcolemma of the muscle fiber, because the acetylcholine will constantly bind to the receptors, and it's going to constantly keep those ion channels open. So you have an enzyme, acetylcholine esterase, which is going to break down the acetylcholine in the synaptic cleft, therefore stopping this signal. So this is a little schematic movie that you should go through and watch that shows the event at the neuromuscular junction. All right, so this is last step 6 is just showing you that the acetylcholine gets degraded by the acetylcholine esterase. Okay, so again, you want the acetylcholine to be removed from the synaptic cleft so it won't stay opening up ion channels in the sarcolemma of the muscle fiber. And as soon as that acetylcholine has been removed, the ion channels are going to close, and then the ions cannot pass. The sodium and potassium can't pass anymore through the ion channels. As I talked about previously, the resting membrane potential inside the cell is negative, so it's around negative 90 millivolts, and outside of the cell is positive. Okay, so essentially, an action potential is going to cause a change in this electrical charge. It's going to cause the resting sarcolemma to go from negative inside the cell to positive, but then it has to come back to resting membrane potential, so back to negative. So it goes from negative to positive, back to negative. Okay, and that occurs in 3 steps: through the end plate potential, depolarization, and repolarization. The end plate potential is what we've been talking about. So remember, acetylcholine gets released from the axon terminal. It goes through the synaptic cleft, and it binds chemically gated ion channels. All that means, the chemical that's gating this ion channel in this case is the acetylcholine. So acetylcholine is a chemical that's going to bind to these ion channels, and they call it gated because once that chemical binds to it, the ion channel is going to open. So when the acetylcholine binds to these chemically gated ion channels, and the chemical is acetylcholine, it binds to these ion channels on the sarcolemma of the muscle fiber, then sodium, the ion channel opens up, and sodium is going to diffuse into the muscle fiber, and some potassium is going to diffuse out. And this is going to essentially result in the interior of the muscle cell to become less negative, more positive. And that's the end plate potential. Okay? So it's become less negative, more positive. That's an end plate potential, and that's a local depolarization. Okay, so that's just locally been in that one area, small area, it's been depolarized. Okay, that's the end plate potential. And this is showing you an end plate potential generated at a neuromuscular junction. All right, so at that end plate potential, if the depolarization reaches a threshold, okay, so an action potential, when an action potential fires, it's either all or nothing. In order for an action potential to fire, it has to reach a certain threshold, okay? So once a certain threshold gets reached, then you have voltage gated. So voltage gated sodium channels are going to open. So once that voltage reaches a certain positive, once you reach a certain positive um, due to the depolarization, then you have voltage gated sodium channels will open. All right, so that charge is what's causing this sodium channel to open, and then you have a large influx of sodium through the channels, and then it triggers an action potential that is unstoppable. So again, action potentials are all or nothing. Once they reach that threshold, they're gonna fire. Okay, below that threshold, if it's a little bit depolarized, but it's not depolarized enough that it reaches that threshold, you're not gonna have an action potential. It's not gonna fire. But once you reach that threshold, it's gonna fire. And then it's going to spread across the sarcolemma from one voltage-gated sodium channel to the next one in adjacent areas, causing that area to depolarize. So this is showing you step 2, depolarization. So again, you had the local depolarization at the end plate, all right, potential, and then once that local depolarization reaches a certain threshold, meaning it gets certain positive, you're going from negative to positive. That's what depolarization is for negative to positive. Then you reach a certain threshold, then you have an action potential fire. Because you're going to open up these voltage gated sodium channels. So these voltage gated sodium channels are going to open, and then sodium's going to rush in, which causes the inside of the cell to become even more positive, okay, when that sodium rushes in, all right? So that's the depolarization step, generating and propagating the action potential. So remember, again, action potentials, they're all or nothing. If that local depolarization at the neuromuscular junction isn't um, intense enough, then it's not going to cause these voltage gated ion channels to open and allow sodium to rush in. Okay, if it's below that threshold, then these voltage gated channels are going to stay closed. But once it reaches that threshold, the voltage gated sodium channels open up and sodium's going to rush in, which causes depolarization and an action potential to be generated and propagate. All right, so in an action potential, so the cell has to go back to its resting membrane potential. So remember, the resting membrane potential is around negative 90. So it goes from negative 90 to about positive 30, positive 30 millivolts when it's depolarized, and then it has to go back to negative 90, and it's going to go back to negative 90 millivolts by repolarization. Okay, so that's the restoration of resting credentials. So your sodium voltage gated channels are going to close, and the voltage gated potassium channels are going to open. So potassium's going to go out of the cell, back to its initial resting membrane potential, and then you're also going to have your sodium potassium pump that's going to pump 3 sodium out and 2 potassium in. It's going to also help restore the resting membrane potential. And you have this what's called refractory period, in which the muscle fibers cannot be stimulated for a specific amount of time until the repolarization is complete. And there'll be a chart that we can show you that was a figure that you can see this, all these different levels and what's going on, you can visualize it better. But just understand that during this repolarization stage, there's a refractory period in which the muscle fiber cannot be stimulated until the repolarization is complete. So this is just showing you step 3, the repolarization. So that you see the sodium, the voltage gated sodium channels are closed, and then the potassium channels are going to open up, which is going to allow the potassium to go out, which is going to help restore the negative resting membrane potential by the potassium going out. Then also you're going to end up with the sodium potassium pumps going to also help this by pumping sodium out of the cell, inside of the cell, and 2 potassium in. 3 sodium out and 2 potassium in. All right, so this is the figure which I showed you about. So you start off at a resting membrane potential at around -90. So then you have at the end of the axon terminal, you have the acetylcholine goes through the synaptic cleft, binds to the sarcolemma. Chemical ion receptors on the other side opens up those sodium channels. Okay, and then at this point you have localized depolarization. Okay, but if that localized depolarization reaches a certain threshold, okay, reaches a certain positive amount of positive inside the internal part of the interior of the cell membrane, then you have voltage gated sodium channels that open up, and that entry allows depolarization to occur. Once that depolarization occurs, all right, then the action potential is going to fire. All right, and then so at this point you have this peak is around +30 inside of the cell. Okay, and now the sodium channels are going to close, potassium channels open. So now we're entering the repolarization phase. So the essentially the sodium channels are now closed, potassium channels are open. So we're repolarizing inside of the cells, becoming more negative, and you're also going to have the sodium potassium pumps going to be helping out the inside of the cell get more negative too. All right, and then you end up back to the resting membrane potential. All right, and this is another short video that you should watch. We've already talked about. So remember, in your motor neuron is coming from the central nervous system. So the nerve impulse is going to travel down the axon until it reaches the axon terminal. And at this axon terminal, you're going to have voltage gated calcium channels are going to open. Okay, and that calcium in the axon terminal is going to cause vesicles full of acetylcholine to be exocytosed into the synaptic cleft. They're going to bind on the other side of the synaptic cleft the sarcolemma, receptors on the sarcolemma, which are going to be chemically gated ion channels, which are going to allow sodium to come in and cause local depolarization. And then if that local depolarization reaches threshold, then it's going to open up voltage gated sodium channels, which is going to initiate an action potential. Remember, action potentials is all or nothing. Once you reach threshold, the action potential is going to fire. Okay, if it doesn't reach threshold, the action potential does not fire. It's all or nothing. Muscle fiber contraction. So you form these cross bridges. So we're going to talk about it in more detail, but essentially the action potential travels along the plasma membrane of the muscle fiber, the sarcolemma. So remember the T tubules are an extension of the sarcolemma. So those action potential is going to travel into the T tubules to the terminal ends of the T tubules, And remember, at these triads, you have in between the T tubules, you have the sarcoplasm. All right, so that action potential, when it travels down the T tubules at those triads where the sarcoplasm reticulum is there in the middle, it's going to cause voltage gated calcium channels to open up, and then you're going to have calcium that's going to leave the sarcoplasm reticulum, and it's going to be able to now bind to troponin on the muscle of the thin filament. So essentially, you have tropomyosin, which is blocking the myosin heads from attaching to the actin thin filaments. Remember, the myosin head is on the thick filaments. So you have this myosin, which is essentially blocking the myosin binding sites on the actin thin filaments. Okay, so what happens is calcium binds to the troponin, which causes the tropomyosin to move and allows the myosin heads to bind to the actin. And essentially, what you're going to end up with is the cross bridge formation first, when the myosin heads, high energy myosin head binds the actin thin filaments in the active site. And then you have the working power stroke when the myosin head pivots and pulls the thin filament toward the M line. And then you have the cross bridge detachment. So the ATP attaches to the myosin head, causing the cross bridge to detach. And the myosin head is going to be left in a high energy state, which essentially is going to be ready for the next cross bridge cycle. And this is a schematic showing you, so you have step number one, the cross bridge formation. So the energized myosin head attaches to the actin myofilament. So you see this tropomyosin, this is that brown long band, is the tropomyosin. And then you have troponin right here, okay? So the calcium binds to the troponin, which causes the tropomyosin to shift, which allows the myosin head to bind to the actin thin filaments, okay? So you're forming your cross bridge. Step number one, power stroke. So ADP and phosphate are released from, so you look up here at step number one, you still have the ADP and phosphate are attached to the myosin head. So the ADP and phosphate are going to be released, and the myosin head is going to pivot. You see how it's pivoting? So it's going to pivot, and it's going to change to its bent low energy state. All right, and as a result, it's going to pull the actin filament toward the M line. All right, and then you have step number three, which is going to result in cross bridge detachment. So ATP is going to attach to the myosin, and that's going to cause the link between the myosin and the actin to weaken, and then the myosin head is going to detach from the actin completely. All right, and then the step number four, the myosin head is going to hydrolyze the ATP, resulting in it to return to its power stroke, or its pre-stroke high energy or cocked position. Okay. So I shouldn't have used the word power stroke. It's going to return to its pre-stroke high energy state at step number four, and it'd be ready to form another cross bridge with the actin. As soon as calcium becomes present, it'll be ready to form another cross bridge. Then it's going to bind to the actin. Then it will go through the power stroke, and then it's going to detach. Then it's going to go back to the cocking of the myosin head when it hydrolyzes the ATP, and then it's going to keep going through this cycle. So another video you should watch. All right, so this is just a clinical note. So we all know from movies that essentially, is no other way to put it, dead bodies stiffen. Okay, so essentially, there's a scientific reason for that. All right, and it's called rigor mortis. But essentially, um, after someone passes away, the intracellular calcium level is going to increase because ATP is no longer being synthesized, so calcium is not going to be pumped back into the sarcoplasmic reticulum. As a result, the ATP is also needed for cross-bridge detachment. So as a result, the myosin heads stay bound to the actin, causing constant contraction. So the muscles stay contracted until the proteins break down. Okay, so essentially that's why the dead body becomes stiff, because the muscles are staying contracted because you don't have ATP present anymore. All right, so this slide, it just has some questions and then answers that you write. So what is the name of the piece of connective tissue that surrounds an individual muscle cell endomysium? The gap between the axon terminal and sarcolemma is the synaptic cleft. In reference to bones for muscle movement, the part of the muscle attached to the immovable bone is called the origin. For action potential to be spread, calcium ions are released that lead to axon terminal's ability to release the neurotransmitter acetylcholine. Once this neurotransmitter is released, the sarcolemma of the muscle cell fiber becomes permeable to sodium. Principles that apply to contraction of a single fiber applies to whole muscles. So contraction produces tension, and the force exerted on load or object to be moved. So tension is the force exerted on load or object to be moved. And this isn't a physics class, but if you remember from physics, forces, they act in pairs. For every action, there's a reaction. So essentially, the contractive produces a tension, all right, that is exerting a force on a load, okay? But that load is also, so think of it this way, the muscle is pushing on the load, but the reaction is the load is actually pushing back on the muscle. So when so you can have two types of contraction. You can have isometric contraction. That means there's no shortening of the muscle. So the muscle tension increases, but it does not exceed the load. So if it doesn't exceed the load, then you can't you can't move the load, okay? If it doesn't exceed the load, all right? And then isotonic contraction, the muscle shortens because the muscle tension exceeds the load, okay? All right. Vary muscle contraction. You can vary in the force and you can vary in the duration of the contraction, which it can cause um, either all like a large area of muscle to contract or a very small area to contract, or the muscles might contract, but it might not shorten in isometric contraction, which serves for posture. So, but all those deal with the force and duration of the contraction. All right, and essentially remember that you have a motor neuron contains axons up to hundreds of motor neurons, and the axons branch to terminals each which forms a neuromuscular junction with a single muscle fiber, and the motor unit is the nerve muscle functional unit. I don't even know what I just said. Consists of the motor neuron and all the muscle fibers it supplies. So a single motor neuron can supplies from anywhere from 4 to several hundred muscle fibers. The smaller the fiber number, the greater the fine control. So essentially, muscle fibers from a motor unit are spread throughout the whole muscle. So stimulation of a single motor unit is only going to be a very weak contraction, all right, of an entire muscle. Which consists of one motor neuron and all the muscle fibers it innervates. So this is a motor unit here, motor unit 1 and motor unit 2. So again, consists of a motor neuron. So this is the motor neuron and all the muscle fibers it innervates. Remember, at the end of the axon terminal, you're gonna have branching off, okay? And then they're gonna branch off, and it's going to bind. It's going to end in a neuromuscular junction, okay? So you're gonna have at these axon terminals, you're gonna have a synaptic cleft, and then you're gonna have the muscle fiber on the other side of that synaptic cleft, okay? So these axon terminals are gonna bind with from a few to hundreds of muscle fibers. All right. It's always easier to study one one thing and then study a larger number of things. So when they study muscle contraction, they like to first start off by just studying a muscle twitch, which is the simplest contraction resulting from a muscle fiber's response to a single action potential from motor neuron. So essentially, the muscle fiber is going to contract quickly, then relax, and the twitch can be observed and recorded as a myogram, which is essentially a line recording contraction activity. All right, so this is a myogram showing the three phases of isometric twitch. Have the latent period, and that's the first few milliseconds following stimulation when the excitation contraction coupling is occurring. So during this time period, you're going to have cross bridge begin to cycle, but muscle tension is not yet measurable. All right, so this is this latent period right here when the muscle tension is not yet measurable, okay? But you're going to have the excitation contraction couplings occurring, but the muscle tension isn't measurable yet. This is that latent period right here, okay? All right, so then next, the second period, you have the period of contraction. So during the period of contraction, cross bridge are active from the onset to the peak of tension development, and the myogram traces rises to a peak. All right, so it's going to rise to its peak, and this lasts about 10 to 100 milliseconds. All right, and then the third step, the period of relaxation. So this is the final phase. It lasts um, about 10 to 100 milliseconds, and is due to the pumping of calcium back into the sarcoplasmic reticulum. So you're pumping calcium back into the SR. All right. So because the number of active cross bridging is declining, the contractile force is declining. Muscles contract faster than it relaxes. So you see how fastly the muscle contract, and you see how slow it is relaxing. Okay, so the muscles contract a lot faster than it relaxes. So the normal muscle contraction is relatively smooth, and the strength varies with needs. So a muscle twitch is only seen in lab setting or with neuromuscular problems, but not in normal muscle. So it is not normal for your muscles to twitch. I know before I got like eyeglasses, I used to read a lot, and when I was reading a lot, my eye would start to twitch. Okay, because my eyes was essentially straining because I needed glasses. Okay, but again, that muscle twitch isn't normal. Once I got my glasses, when I read, okay, I didn't have the muscle twitch in my eye anymore. Okay, so normal muscle contraction is smooth. Okay, and it graded muscle response varies strength on contraction for different demands. So required for proper control of skeletal movement, and the response are graded by, you can change the frequency of the stimulation. The greater the frequency, the greater the stimulation, or you can change the strength of the stimulation. So muscles respond to changes in stimulus frequency. So single stimulus results in a single contractile response. A muscle twitch, for example, and muscle respond to the changes and stimulation frequency. This is a single stimulus, a single twitch. So you see the stimulus, it contracts, and then it relaxes. Okay, so in a single, when a single stimulus is delivered, the muscle contracts and it relaxes. But if the muscle is stimulated at an increasingly faster rate, what's going to happen is the relaxation time between the twitches becomes shorter and shorter, and the concentration of calcium in the cytosol is going to rise higher and higher, and then you're going to have waves are going to start to add together. Okay, so the waves are going to get bigger because they're going to start to add together until you end up with a essentially a completely fused um, plateau um, graph. So this wave summation results of two stimuli received by a muscle in rapid succession. So the muscle fibers don't have a complete time to relax, and so it's going to increase in force with each stimulus. All right. Then you also have additional calcium that's being released with the second stimulus, which is going to stimulate more shortening. So it's going to produce. You continue this if you keep adding stimulus. So if you keep adding stimulus, increasing the frequency of these stimulus, it's going to produce smooth, continuous contractions that add up. Okay, they're going to sum up. And if you still complete go on continuing the stimulus, it's going to become a sustained quivering contraction referred to as an unfused tetanus. Showing you temporal summation. So essentially, the additional stimuli delivered before the relaxation occurs produces this temporal summation. All right, and if you continue with this temporal summation, what's going to happen is that the wave summation will become greater and greater, so you end up with a sustained but quivering contraction that's called an incomplete tetanus. Okay, and then if you keep applying more and more stimuli, if you keep adding more and more stimuli, then you end up with a fused complete tetanus. And in reality, your muscles never actually ever reach a complete tetanus because you're gonna end up with muscle fatigue. So your muscles will give out before you actually get to a complete tetanus, um, a fused complete tetanus. And this is showing you a fused complete tetanus. All right, you see how you have this plateau. All right, and your muscles are completely being contracted, and again, this they can't sustain this, so you're gonna end up with muscle fatigue. This is not sustainable. And there's essentially no relaxation between the stimuli. All right. Changes in stimulus strength. So you essentially have what's called the recruitment works on size principle. So the motor units with the smallest muscle fibers are recruited first, and then you have motor units with larger and larger fibers are recruited as the stimulus increases intensity, and the largest motor units are activated only for the most powerful contractions. So you have this asynchronous muscle contraction, essentially. So some fibers are contracting while others rest, and that helps prevent muscle fatigue. So this is just showing you the size principle of recruitment. So you always start off with the smallest muscle fibers, um, then you get medium fibers, then you get the large fibers get activated last. So you have what's called posture. So to keep your posture, you actually have a constant, slightly contraction state of all your muscles due to spinal reflexes. All right, so that doesn't cause your muscles to move. It just keeps your posture. All right. So groups of motor units are alternately activated in response to input from your stretch receptors and muscles. So it keeps your muscles firm, healthy, and ready to respond. So it's important. So with isotonic contractions, the muscle changes in length. Oh, I should have mentioned that with your posture, to keep your posture and your tone, these are going to be isometric contractions. Your isometric contractions are what's going to keep your tone, your posture. Isotonic contractions are going to result in changes in muscle length and muscle load. So they can either be concentric or eccentric. So with the concentric muscle contractions, the muscle going to shorten and does work. Example, you picking up a book. Eccentric contraction, the muscle's going to actually lengthen and they generate force. Okay. So example, laying a book down causes the biceps to lengthen while generating force. Okay. So again, these are both concentric and eccentric, are both isotonic types of isotonic contraction because one is shortening the muscle and one is lengthening the muscle, generating a force. Okay, and the other one is shortening the muscle and it does work. All right. An example of concentric isotonic contraction. So you have this weight on this muscle and the muscle contracts and it's doing work. It's when it's contracting, it's shortening, but it's also doing work by lifting up this um, by moving this weight. So with isometric contractions, remember the muscles don't contract. So the load is greater than the maximum tension the muscle can generate. So the muscle neither shortens nor lengthens. Okay. So isometric, the contractions, the muscles don't contract, they don't shorten, they don't lengthen. So electrochemical and mechanical events are the same in isotonic and isometric contraction, but the results are different. So with isotonic contraction, actin filaments shorten and cause movement. In isometric contraction, cross bridge generate force, but actin filaments do not shorten. So essentially, the myosin head just kind of spin their wheels on same actin binding sites. So this is showing you isometric contraction. So essentially, the force that the muscles generate isn't greater than the load. Okay, so the muscles can't shorten. If they shorten, that means they would be lifting the load. In order to lift the load, the force of the muscle has to be greater than the load. So essentially, think of it as, remember I said with physics, for every action is a reaction. So when I push my arm against the wall, so my hand is pushing a force against the wall, but also the reaction is the wall is pushing a force against my hand. And in this case, the wall has a greater force than the force that I can generate pushing against the wall, so the wall doesn't move. Okay, it's the same thing for this muscle that can't lift this weight. Okay, it's not generating a force greater than the um, force generated by the weight. So the muscles can't shorten because it can't lift the load. Energy needed for contraction. All right, so contraction needs a lot of energy. So a lot of the processes that we talked about need ATP. So remember those calcium pumps need ATP to pump the calcium back into the SR. Sodium potassium pump requires ATP. Remember when the myosin head is attached to the actin, ATP has to bind to it, which causes it to detach and return back to its high energy state after the ATP gets hydrolyzed to ADP. So in order for that, the cross bridges to detach, ATP has to bind to the myosin head when it's bound to actin. Okay, so you need energy for this entire process, and essentially, the available stores of ATP are depleted in about 6 seconds. And ATP is the only source of energy for contractile activities. So it has to be regenerated. So let's talk about different ways that we can regenerate ATP for our muscles. All right, so you can either do 3 ways: direct phosphorylation of ADP by creatine phosphate, anaerobic pathway through glycolysis would generate lactic acid, and then aerobic respiration. All right, so one of the fastest ways to generate ATP is going to be using creatine phosphate. So it's a unique molecule that's located in muscle fibers that's going to donate a phosphate to ADP to form ATP. So it uses the enzyme creatine kinase to carry out the transfer of the phosphate. So muscle fibers have enough ATP and creatine phosphate reserves to power cell for about 15 seconds. So this is going to be um, a fast reserve. It's just to use creatine phosphate, okay? And it's a molecule that's going to be located in the muscle fibers, and it's going to be there just to donate a phosphate to ADP to form ATP. So again, this is going to be a fast way to generate more ATP. So this is just showing you creatine phosphate, and this is showing you ADP. Creatine kinase is going to be the enzyme that catalyzes the transfer of a phosphate from creatine phosphate to ADP, forming ATP and resulting in creatine. And just a side note, I remember this is long time ago. It was like when I was in high school, but there was this big creatine phosphate was this big new supplement that everybody was taking to give them more energy to work out. So that's just kind of a side note, but there is some biochemistry to taking creatine phosphate. But again, it's a molecule that's found naturally in your muscle cells, and it's used to donate a phosphate to ADP in order to form ATP quickly. Okay. All right. So normally your muscles want to use, like I said, they can use the creatine phosphate real quick. That's a fast way to generate ATP. But also your muscles want to normally, if they can't use, once they use up the creatine phosphate, they normally want to use the aerobic pathway. Okay. And we're going to talk about why the aerobic pathway is better than the anaerobic pathway. Essentially, just the aerobic pathway generates more ATP. But the anaerobic pathway, so say you're working out really, really hard, and you can have what becomes oxygen can become deprived in your muscles if you're working out really, really hard. You don't have enough time. Your muscles are contracting, and you don't have enough time for your muscles to relax, to allow oxygen to flow through. Okay, so you can end up with essentially an anaerobic environment for your muscles when you're working out really, really hard in that state. And essentially, the only option your muscles have at that point is to use the anaerobic pathway, and that's going to be through glycolysis. And we haven't talked about in a lot of detail. We mentioned when we talked about the mitochondria how ATP is generated, but essentially the first step of ATP generation is glycolysis. So glycolysis just means the glucose gets broken down to two pyruvate molecules. So when the glucose gets broken down to two pyruvate, so glucose has 6 carbons, pyruvate has 3 carbons. So the glucose is going to be broken down from a 6-carbon molecule to 2 3-carbon molecule, okay? And that's glycolysis. All right, and through glycolysis, that happens in the cytoplasm of the cell, and it does not require oxygen. Glycolysis does not require oxygen, and essentially you produce about 2 ATP. All right, so it doesn't generate much energy. Now here's the issue. Okay, without oxygen, you can't. The two pyruvate that's generated, they can't go to the mitochondria. Okay, in order for the mitochondria to function, you need oxygen, because in the electron transport chain, which is in the mitochondria, oxygen is the final electron acceptor. So essentially, what you end up with anaerobic respiration, you end up with two pyruvate that essentially what has to happen if the pyruvate can't go into the mitochondria and be broken down even more, okay, in the mitochondria, the pyruvate is broken down to CO2, and then that CO2 goes off as gas to be broken down even more, and then in the mitochondria and through the electron transport chain, you generate like 32 ATP. If you can't do that, then what happens to the pyruvate? In order for glycolysis to continue, so essentially, you generate electrons when you go through glycolysis, but you need somewhere to donate those electrons. So the pyruvate can become the electron donors, and through the process of the pyruvate becoming the electron donors, um, so that glycolysis can continue. The pyruvate is converted to lactic acid. Okay, so lactic acid is like a byproduct result of from the pyruvate becoming the electron acceptor, so that glycolysis can continue. Because you have enzymes um, through in glycolysis and enzymes in aerobic respiration that essentially transfer. They go through these oxidation reduction reactions, and electrons get transferred from one molecule to another, and you need these electron acceptors. Okay, and that's why I said why you need oxygen for the aerobic respiration, because oxygen is the final electron acceptor, and oxygen becomes water when it accepts the electron, and that's one of the byproducts of aerobic respiration, water, and it gives off CO2. But with glycolysis, in order to keep glycolysis going, so you break down glucose and you generate 2-pyruvate, but in order to keep that going, you need an electron acceptor for glycolysis too, and pyruvate can accept electrons, and in the process, when pyruvate accepts electrons, it's going to be transformed into lactic acid, okay? And then now you're going to end up with lactic acid being built up, and there are several ways that your body deals with the lactic acid. It can diffuse into the bloodstream, and your liver can convert it back to pyruvate. And other cells can use lactic acid as energy too. So certain things have to lactic acid, but again, this anaerobic pathway isn't sustainable long term because you just don't generate enough ATP through it. But again, it can serve long enough until you can essentially stop exercising and you can get rid of your oxygen deprivation. Essentially, you don't have enough oxygen until you can regain your oxygen that you need so that you can do aerobic respiration. All right, so this is just going into more detail what I got through talking about. Normally, pyruvate enters the mitochondria to start aerobic respiration, but at high intensity activity, the oxygen is not available. And again, you can't do aerobic respiration without oxygen because oxygen is the final electron acceptor. And when it accepts the electron, it becomes water. So you can't do aerobic respiration without oxygen. But bulging muscle compression blood vessels, so you have impaired oxygen delivery. So in the absence of oxygen, you have to do anaerobic glycolysis, and the pyruvate gets converted to lactic acid. So essentially, the pyruvate becomes the electron acceptor, and in that process, it gets converted to lactic acid. All right, so then that lactic acid can diffuse into the bloodstream. It can be used as fuel in the liver, kidneys, and heart, and it can be converted back to pyruvate or glucose by the liver. Okay. So anaerobic respiration yields only about 5% the ATP as aerobic respiration, but it does it faster. It does it 2.5 times faster. Okay. So this is just showing you the anaerobic pathway. So remember, glucose enters a cell. Glucose gets broken down through glycolysis to pyruvate, okay? And it generates 2-ADP. Normally, this pyruvate would go into the mitochondria if oxygen is present. But when oxygen is not present, what happens is pyruvate becomes the electron acceptor, and it gets transformed into lactic acid. And then that lactic acid gets released into the bloodstream. All right, so with aerobic respiration, you produce 95% of the ATP during rest and light to moderate exercise. It's slower than the anaerobic pathway. And essentially, you have like 2 steps in the mitochondria. First, you got to go through the Krebs cycle, or sometimes it's called the TCA cycle. okay? And through that TCA cycle or the Krebs cycle, the pyruvate is going to be broken down into CO2. So it can keep being broken down until you give off CO2. And then you're going to generate a lot of electron carriers that are going to carry the electrons to the electron transport chain, which is still in the mitochondria. And then in the electron transport chain, those electrons get passed down and passed down and passed down the chain until they reach oxygen, which is the final electron acceptor that generates the water. Okay, so between those two processes, you can produce around 32 ATP. All right, so this fuel includes glucose from glycogen stored in muscle fibers, then blood-borne glucose and free fatty acids. And just note that fatty acids are the main fuels after 30 minutes of exercise. So this is showing you the aerobic pathway. So again, normally what happens, glucose comes to a cell, and the cytoplasm, it goes through glycolysis. Glycolysis breaks down the glucose into pyruvate, two pyruvate. Okay, pyruvate is just a 3-carbon molecule, so it produces 2 of them. Remember, you start off with glucose, 6 carbons. You produce 2 of these pyruvate, which are 3 carbons each. They go into the mitochondria, and then you go through the TCA cycle, and then you go through the electron transport chain. Okay, so after you go through the TCA cycle, you go through the electron transport chain, where oxygen is the final electron acceptor, and you generate water. And the CO2 is really generated through the TCA cycle. All right, and it's just showing you different fuels that you can use. You don't have to just use pyruvate. You can also use fatty acid as a fuel, and even amino acids as a fuel. Okay. All right, so energy systems used during sports. So you have aerobic endurance. So length of time muscle contract using aerobic pathways. This is going to be your light to moderate activity, which can continue for hours. And then you have the anaerobic threshold, so the point at which the muscle metabolism converts to an anaerobic pathway. Okay. So you see right here this nice casual jogging. Okay, so this is good. This is going to be aerobic, but you see this short duration, high intensity sprint. This is going to result in using at the end anaerobic pathway. Okay, but you start off, you're going to use the creatine phosphate first. Okay, because that's always a good fuel first. You have enough ATP stored for about 6 seconds. After the 6 seconds, you're going to use your creatine supply. After your creatine phosphate donated all the phosphate that it has to the ADP to make ATP, then you have to go through the anaerobic pathway. Okay, and you can use glycogen stored in muscles for that to get the glucose, which can be used in the anaerobic pathway through glycolysis to generate ATP. All right. And then, but if you're just doing a prolonged, nice, leisurely jog, you can use the aerobic pathway, okay? The entire time, essentially, with the nice, leisurely jog. All right, so you can have what's called muscle fatigue. So muscle fatigue is when the muscles can't contract even though they have stimulation. So that can occur when there are ionic imbalances. So levels of potassium, calcium, and phosphate can interfere with the EC coupling. Prolonged exercise may also damage the SR and interfere with calcium regulation and release. So lack of ATP is rarely the reason for fatigue. All right, so just understand, usually it's not because there's not ATP available. Okay, so it's usually something else. Prolonged exercise may damage the SR and interfere with calcium regulation and release, okay? Or the levels of potassium, calcium, and phosphate can interfere with EC coupling. But normally muscle fatigue is going to be caused by these reasons, and that can be caused because lack of ATP, all right? There's going to be ATP there. That's not going to be the problem. The problem is going to be that the muscles are fatigued and they can't contract even though they're being stimulated, all right? All right, so after you get through exercising, if you had essentially was using anaerobic pathway, meaning that there wasn't enough oxygen getting to your muscles, you're gonna be in a state that's gonna have to replenish your oxygen reserves. Okay, and your lactic acid that you have built up can have to be reconverted to pyruvate, and the glycogen stores gonna have to be replaced. Okay, and ATP and creatine phosphate reserves have to be resynthesized. So all these things have to happen post exercise when you were in an anaerobic state. Okay, because you had essentially not enough oxygen, so you got to go replenish all your stores of oxygen. You have to get rid of the lactic acid. You got to replenish the glycogen, and you got to regenerate, resynthesize ATP and creatine phosphate. And this is called the exercise post exercise. I'm sorry, excess post exercise oxygen consumption (EPOC), formerly referred to as the oxygen debt. All right, we're gonna stop here for this lecture. So contraction, and then we'll go into some talking about the functions of different muscles in our body in the next lecture.